Okay, so we do have a solution. We can use something like DEXA. We can quantify fat-free mass, and we can now express VO2 max relative to fat-free mass. And what we find is, in this case, where males started with this huge advantage in absolute VO2 max, the difference is roughly 0. So it is possible to match males and females who have similar training histories for VO2 max. Okay, this, as it turns out, is the gold standard for matching males and females. It works because now this measure of VO2 max is unrelated to size. It doesn't matter if you weigh 40 kilos or 80 kilos, your VO2 max isn't depending on that variable anymore. It turns out if you look at a number of, I guess, either components of body mass or ways to compartmentalize body mass, as long as you remove fat mass, it doesn't really matter. This measure doesn't relate to total lean mass if you take away bone, leg fat-free mass if you're just trying to isolate the most active tissue, trunken leg if you're trying to account for some visceral organs. It seems like as long as you remove fat mass, you can compare fitness for males and females in a way that makes sense. Okay, we wrote this review in 2025, but it's not a new idea whatsoever. This paper is from 1957, and their conclusion is, kind of summarizing what I've just said, the presence of excess fat per se does not have any important influence on the capacity of the cardiovascular system to deliver oxygen under maximal conditions. So that's 78 years ago. Slightly more recent, this is now we're 1981, I'm still not alive, but do you have people saying, okay, this is the probably easiest and most valid expression to use for the matching of male and female subjects? It takes into account both differences in size and body composition. But from 1981 until now, no one is really pushing for this to be done. You'll find studies that do it, but most studies kind of just ignore it. And so we've tried to do this in all of our studies in my 8 years here, and we have numerous studies that have used this approach. Rachel will talk about the most recent one. But before we get there, a question you might have, does this mean males and females are equally fit? No, it doesn't. If you just recruit participants without any specific recruitment strategies, I need a fitter male to balance this out, I need a less fit male, what you will find is males will still tend to have a greater VO2 max per fat-free mass. So there are still population differences in fitness. So what we're not saying is globally males and females are equally fit. We're saying we can create samples where fitness is matched, and now we can say in males and females who are matched for fitness, is there a difference in some other variable removing that effect of fitness? So we know things like physical activity and diet influence fitness, and we know gender and sex influence physical activity and diet. So don't leave here thinking I said males and females are equally fit everywhere. That's a much messier question that we can't really answer in our lab. But when we do match, now we can say, is there a sex difference in something like hemoglobin mass? So hemoglobin mass, really simply, is just if you took all the hemoglobin protein out of your body and plopped it down onto a scale, what would it weigh? We don't do that as a method. We have people breathe carbon monoxide, and we measure how much carbon monoxide is in their blood before and after breathing. And by the increase in carbon monoxide bound to hemoglobin, we can use the dilution principle to estimate how much hemoglobin they have. And so if we do this, we get a distribution like this. So females still in purple, males in green. You can see two overlapping distributions, different means. If I express it this way, you can see they're almost barely overlapping. In general, males have a lot more hemoglobin than females. Now, it's important to point out in this study, these males and females are matched for fitness. So this is a group of male and females. We've slightly changed their sample size. Not everyone in the last data set has this data, so this is, think of it as a subset. But these groups of males and females have the same average VO2 max per fat-free mass, but males have 50% more hemoglobin. So it's not enough just to match the groups. We still need to correct for differences in size and composition for our variable of interest. So again, this is a low-hanging fruit. I can easily, without much data, say males have more hemoglobin mass than females. What I want to know is, do they have more hemoglobin mass for their size than females? And the key here is the relationship between fat-free mass and hemoglobin mass is very strong. It's, of everything we've measured, it's the strongest predictor of your hemoglobin mass. And again, now in the subset, we still know males are larger than females, particularly when we look at fat-free mass. So if we correct their hemoglobin mass for this fat-free mass, is there a sex difference? And what we find is yes, and it's still significant, it's still relatively large, but it's not 50%, it's 9% in grams per kilo. So the average female that we've studied has about 14 grams per kilo, the average male about 15 grams per kilo, which is a much smaller difference than, say, 650 versus 950. So this would be what I would call a true sex difference. This difference isn't because there's a difference in fitness, because with training, anyone can increase their hemoglobin mass. This difference, not because males are bigger, this is what we get once we've matched for fitness and controlled for size. Okay. Quickly, I just want to show you what would happen if you didn't do this. This slide is a little busy. The first version of this figure was much busier, so I'm proud at how simple it became, but I appreciate it is not that simple. If you look in the center of this figure, what we have is if we match males and females for VO2 max per body mass, we will get females that are fitter than males. So this is what we want to do. We want to match for fat-free mass and let males look fitter, good for their ego. And that's what shows us that we probably did it right. So if you can't measure body composition, this is what you're aiming for. Males who are slightly fitter per body mass is roughly matched for fat-free mass. But if we do it the other way, if we don't have a DEXA or some way to quantify fat mass, we'll get females fitter than males. Then when we go to our variable of interest, and let's say we're using hemoglobin mass as an example, where males have a greater value, this is where it gets a little complicated. But now, instead of having the same VO2 max per fat-free mass and saying males have a higher value than females, we actually have a higher VO2 max per fat-free mass. So when we go up here, we get an inflated value for females, which I call a fitness effect. And the result of that study would be there is no sex difference. So if we match for VO2 max per body mass, we can falsely conclude that males and females have the same hemoglobin mass per kg, when we know they absolutely do not. So that's more of a caution of if you see it match that way, be suspicious. Okay, that is my long intro for Rachel's study. To recap, in exercise physiology, when we compare sexes, we often have to match for aerobic fitness. Not every variable every time, but often. With endurance exercise, related parameters, the best method is VO2 max per fat-free mass, and most variables of interest still then have to be scaled to account for differences in size and composition. Okay, so I will now seamlessly transition to Rachel presenting. And she will show you her thesis.
Okay, I am just getting over a cold, so please bear with any raspiness that comes through. Okay. So, yeah, thank you. I'm gonna be talking about my master's project, which I just defended like 2 weeks ago. And it was looking at the influence of sex on critical power during dynamic knee extension. Okay. High intensity exercise performance can be modeled by a hyperbolic relationship between work rate and the duration it can be sustained. So this relationship models performance using 2 parameters.
That works.
Barton, did you break it? It's not.
I don't remember how I was changing slides.
Oh, now it's working. How'd you do that?
Very strong.
Okay, the critical intensity is the horizontal asymptote of this relationship, and it represents an intensity theoretically sustainable indefinitely. It's the threshold between the heavy and severe exercise domains. So the critical intensity is kind of an umbrella term, and it can be made more specific to the testing modality. So the critical intensity is often measured as critical power, critical torque, critical speed, etc. W prime is the curvature constant of this relationship, and it represents a finite capacity for an individual to complete work above the critical intensity. So some sex differences, as Martin has talked about, have been established in exercise physiology research. So males are generally equipped to achieve a higher absolute work rate. They're larger, they have larger total muscle mass, larger lungs and airways, and a higher hemoglobin mass. On the other hand, females may be better equipped for oxidative metabolism. With a greater proportion of type 1 fibers, this may allow them to achieve higher rates of mitochondrial respiration and greater capillarization in the muscle. Some of these differences are still contested in the literature, but they bring on the question, are there sex differences in endurance performance? And specifically today, we're going to talk about, are there differences in the critical intensity? So there have been a few studies over the years that have looked at this. So this is a study that was done in our lab, looking at sex differences in the critical intensity as critical power for whole body or cycling exercise. So in this case, as Martin talked about, super important, our males and females were matched for relative aerobic fitness, so VO2 max expressed relative to fat-free mass. So in these fitness matched males and females, males achieved a higher absolute critical power, but that difference was reduced when critical power was expressed relative to body mass and reduced further when expressed relative to fat-free mass. So we see in these fitness matched males and females, there was no sex difference in critical power for whole body exercise. Since females are said to have a more oxidative muscle profile, what happens if we look at the critical intensity using a more isolated test? Knee extension exercise is often used as a small muscle mass modality to isolate the quadriceps and remove the effect of central factors, so factors relating to oxygen delivery to the muscle. And this gives us a way to effectively compare muscle performance. So this study done by Ansdell and colleagues, they used isometric knee extension to compare the critical intensity as critical torque between males and females, and they found that females had a higher relative critical torque than males. However, it's important to point out that the males and females were not matched for aerobic fitness in this study. So there's no way for us to confirm that the sex difference they saw was truly a difference due to biological sex and not relating to a fitness difference between groups. This is another study done by our lab, which, similar to the Ansdell study on the previous slide, used isometric knee extension to investigate sex differences in critical torque. But here, as you can see in that top right corner, our males and females were matched for VO2 max expressed relative to fat-free mass. And so when matched for fitness, unlike the Ansdell study, no difference was seen in critical torque, although males demonstrated a numerical advantage for absolute critical torque on the left there, and females had the numerical advantage for critical torque relative to maximal voluntary contraction on the right. But neither of these differences were significant. So even though implementing appropriate fitness matching is progress in the right direction, both of these studies failed to account for differences in lower limb anatomy between sexes and the biomechanical consequences of these differences. So one important thing, torque is equal to force times the moment arm. So we can say that quadricep driven knee extension torque is equal to quad force times the patellar tendon moment arm. Males generally have a longer moment arm than females, meaning that they require less force output from the muscle to generate a given knee extension torque. So to kind of sum up all of the previous work, progress has been made in studying sex differences in the critical intensity, but findings have been mixed, and there are still some limitations that need to be addressed. And today we're focusing on single leg exercise, so that small muscle mass exercise. So in addition to implementing fitness matching and accounting for anatomical differences between males and females, it's also important to look at dynamic exercise, which is generally more reflective of activities you would see in sport and daily living than isometric exercise. So that brings me to the primary objective of this study, which was to look at sex differences in the power duration relationship for dynamic knee extension exercise. We also wanted to look at potential determinants of this relationship, including aerobic fitness, muscle oxidative capacity, and muscle strength. We hypothesized that males would achieve a higher absolute critical power, but after controlling for muscle mass, that there would be no difference in critical power, or specifically the torque at critical power. That screen just flashed off. Yeah, we predict that there would be no sex difference in torque at critical power after controlling for muscle mass. However, we predicted that females would require a greater quad force at critical power to overcome their structural disadvantages and produce similar torques to males, given their shorter patellar tendon moment arms. We also hypothesized that aerobic fitness and muscle oxidative capacity would be related to critical power, whereas muscle strength would be related to W prime as the anaerobic parameter. So we recruited 12 males and 12 females to investigate these questions. This is an overview of our experimental design, and it was broken into 4 stages taking place over the course of 7 lab visits. So participants first came in for measures of body composition, strength, rate of force development, as well as familiarization to study protocols, and then VO2 max tests to assess maximal aerobic fitness for both whole body, so cycling, and single leg exercise. Next, we completed critical power testing involving 4 constant work rate trials over the course of 2 visits. Then we moved on to NIRS oxidative capacity, and finally our MRI of the knee and thigh. So for our whole body fitness assessment, we used a maximal ramp incremental test on a cycle ergometer using a metabolic cart to measure gas exchange data, and specifically we were looking to assess VO2 max. And so again, as Martin has already shown, although males achieved a higher absolute VO2 max than females, this difference was reduced when VO2 max was expressed relative to body mass, and the difference was virtually eliminated when expressed relative to fat-free mass. So normalizing to fat-free mass, it corrects for those differences in body size and composition between males and females, and it shows that our males and females were matched for maximal aerobic fitness. So this means we could go ahead and compare muscle performance and attribute any differences we saw to biological sex rather than fitness. So the rest of the testing was conducted on a new apparatus, a dynamic knee extension ergometer. Participants performed knee extensions with their foot secured in a snowboard boot style binding attached to a crank arm, and the crank arm rotated the flywheel with each extension that they performed, and they kind of just did this continuously, almost analogous to pedaling a bike. And they were instructed to allow the flywheel's momentum to passively return their foot back to the starting position between each extension. So this figure is showing a representative example of data from the ergometer over 5 full contraction cycles at a fixed cadence of 40 RPM. So this is our instantaneous force and power output data. We added an additional trace to correct for a few shortcomings of the raw power output data we got from the machine. And this bottom trace was what was used in all of our analyses. So first of all, we only wanted to account for power where a positive or anterior force was measured by the ergometer to focus on quadricep-driven work. We also wanted to account for any work done by the quadriceps eccentrically or against the direction of the flywheel and add it to rather than subtract it from the total power output. For our critical power determination, participants completed 4 constant work rate trials to failure, ranging from 2 to 15 minutes in duration to ultimately establish the power duration relationship and determine critical power and W prime. NIRS oxidative capacity can provide a non-invasive estimate of mitochondrial content, and here NIRS was used to monitor the relative amounts of oxygenated and deoxygenated hemoglobin and myoglobin to look at muscle oxygen utilization after exercise. So participants exercised for 1 minute at 20% below their critical power, and this was followed by a series of 5-second arterial occlusions ongoing for several minutes. So the change in deoxygenated hemoglobin and myoglobin during each occlusion was measured and plotted. And we then fit a curve using this equation to ultimately determine tau, which was our measure of oxidative capacity. So a higher oxidative capacity reflects a faster metabolic recovery or a faster return back to baseline, which is represented by a lower tau value, lower time constant. For our MRI, we imaged from the top of the iliac crest to just inferior to the tibial plateau to capture the entire quadricep and knee regions. To measure quadricep volume, we used the ideal IQ sequence in the axial plane, and we performed manual segmentation and interpolation to determine a 3D volume. To measure the patellar tendon moment arm, we used the T1 FSE sequence in the sagittal plane, and in that sagittal plane, we measured the distance between the tibiofemoral contact point and the line of action of the patellar tendon. Using our measured values, we performed a simplified estimation of quad force output. So first, we calculated the torque at critical power by multiplying the measured force at critical power by the length of the lower leg. That gave us torque at the knee. So once we knew the knee extension torque at critical power, we could calculate the quad force required to produce that torque. So we divided torque by the patellar tendon moment arm to get quad force. And so, as you can see up here, this calculation was obviously applied for critical power, but we also applied it for maximal strength using the maximal voluntary contraction, or MVC, to determine MVC quad force as well. So getting into our results, starting off with raw values, males achieved a higher critical power as we predicted, and this held true in terms of power output, torque, and quad force at critical power. However, we wanted to account for differences in lower limb anatomy, as males had a higher quad mass than females, and quad mass was related to critical power in the overall sample, so we wanted to include it as a covariate in our analyses. To do that, we used ANCOVA to look at the relationship between quad mass and critical power and compare that relationship between males and females. And so this relationship was not different for power output, torque, or quad force at critical power. Using ANCOVA also allowed us to estimate adjusted means for each sex. And so this was evaluated at a constant quad mass at the mean of the whole sample. So in this case, 2.4 kilograms of quad mass. And our adjusted means were not significantly different for power output, torque, or quad force at critical power. Similar to what we saw for critical power, males achieved a higher absolute W prime than females. And this was also true for maximal strength in terms of both MVC torque and MVC quad force. The ANCOVA showed a slightly different story than what we saw for critical power. We saw an interaction between quad mass and sex for W prime, meaning that this relationship differed by sex. So males showed a positive relationship between quad mass and W prime that was not present in females. And a similar interaction was seen for MVC torque, again with males showing that positive relationship. This interaction means that we can't look at the overall effect of sex because the effect of sex differs across the range of quad masses. Although it looks very similar, there was no significant interaction for MVC quad force, meaning that we could compare our estimated means after adjusting for quad mass. And again, we saw no significant difference there. So what does all of this mean? We saw no sex difference in critical power after controlling for quad mass. And we saw a general agreement in our outcomes between power output, torque, and quad force. And so this suggests that that there was no significant effect of the sex difference in the patellar tendon moment arm on critical power, or that the effect was just too small relative to the variability in critical power torque to see a sex effect for quad force. For W prime, quad mass was related to W prime in males, but not females. And this suggests a greater capacity to translate available muscle mass to external work in males. And it's just speculation, but this may be explained by a greater proportion of type 2 fibers and the resulting advantage in glycolytic capacity for the males. In terms of maximal strength, quad mass was related to MVC torque in males, but not females. And we did not see this significant interaction for quad force. So together, this suggests that the larger male moment arm provided a structural advantage for torque production per kilogram of muscle. And this may have enhanced the torque mass relationship in males. Okay, so moving on to potential determinants of the power duration relationship. This heat map shows Pearson correlation coefficients for relationships between potential determinants of the power duration relationship and its two parameters, critical power and W prime. And stars represent significant correlations here. And so they're shown as both bivariate raw correlations as well as partial correlations controlling for quad mass. And I know there's a lot going on here, so I just want to draw your attention to a few of them specifically. So the patellar tendon moment arm was not related to critical power or W prime after controlling for quad mass. So like we saw for strength, this suggests no independent influence of the patellar tendon moment arm beyond its covariance with quad mass. Whole body and single leg aerobic fitness were related to critical power independent of quad mass, and this suggests that aerobic function truly accounts for some variability in single leg critical power. Nearest derived oxidative capacity was not related to critical power. This result was surprising. It conflicts with previous studies from our lab that found oxidative capacity to be related to the critical intensity for both cycling and isometric knee extension exercise. It's unclear exactly why it was different in this study. It may come down to a modality-specific limitation to critical power other than mitochondrial content. It may be a result of how differences in how results were normalized across studies, or potentially a combination. Oxidative capacity was related to whole body VO2 max, relative VO2 max, and this finding was consistent with previous studies from our lab in both direction and magnitude, and this gives us confidence in our measure of oxidative capacity in this study. MVC quad force was related to W prime independent of quad mass. This suggests that the maximal force generating capacity was related to the anaerobic work capacity. There does seem to be a bit of a trade-off between aerobic and anaerobic capacities, and this is shown by negative correlations between aerobic fitness and W prime appearing after controlling for quad mass, which is related to both. So although this study does represent some more progress in the right direction, there are still some limitations that need to be addressed, and we're currently working on implementing a few of these, and we plan to update our analyses accordingly. But to calculate joint torque, we only measured external force from the ergometer, but it's also important to account for the internal work required to move the limb against gravity and inertial forces. Our calculations assumed that the measured force from the ergometer was entirely perpendicular to the lower leg, but the angle here should be considered to calculate the proportion of that force that is actually perpendicular. And future studies should also account for antagonist muscle co-contraction when calculating total joint torques. To further refine estimates of muscle force, it's important to note that the patellar tendon moment arm changes throughout the range of motion, and more research is really needed in this area to find a way to accurately estimate the instantaneous moment arm throughout exercise to get a better picture of how muscle force changes throughout the range of motion. So to wrap it all up, males demonstrated a higher absolute critical power than females, but after controlling for quad mass, there was no sex difference in torque at critical power. Females did not demonstrate a greater quad force at critical power, suggesting that the patellar tendon moment arm didn't impose a significant disadvantage for the females at critical power. Aerobic fitness was related to critical power as we predicted, although muscle oxidative capacity was not, and muscle strength was related to W prime. So overall, the findings of this study deepen our understanding of the intensity duration relationship for small muscle mass exercise, and they highlight the importance of accounting for anatomical sex differences when comparing performance. So yeah, I just want to say a quick thank you to, first of all, my supervisor, Martin, and everyone that was a part of this project. So thank you.
Floor is now open for questions. And who would like to start us off?
Yeah. Jeremy. In your results, we show like the jitter plot, so it's like all the like move the air bond and all the.
Sorry?
In the last of your results, it showed like the jitter plot, so like the air bond and all the stat plots. Is a one male unit that's like seems to like sit way out on the edge. Is there any results that might have like surprised you, and where that like influential data point might be problematic?
Yeah, so with
Not those.
yeah, yeah, so there is a bit of an outlier, but I think with a lot of these, like that relationship ends up staying very similar without the outlier as well. But yeah, it was a true outlier, and so we didn't see a justified reason to remove it in this study, but yeah, it's a good thing to consider for sure.
Yeah.
All right, so thank you for your talk, and Martin. I actually had a question about this slide because I didn't follow the one piece, so I'll just get you to
Yeah,
reiterate. So you mentioned that you created adjusted means based on the group mean.
so it's essentially what the ENCODE was doing is pretending everyone had the same quad mass. So pretending everyone had a quad mass at the group mean, which in this case was 2.4 kilograms. What would the male, like for example, MBC quad force be, and what would the female be?
Okay, but none of the females had a quad mass of that, and none of the males did. Would it be more appropriate to adjust to the sex specific mean to then get a better understanding?
Potentially, yeah,
I was just wondering if that, the rationale for doing a group as opposed to the sex.
yeah, statistically, I think we were hoping to just use a regression-based analysis, so we looked at ANCOVA to kind of just avoid the traditional route of just ratio scaling, so looking at it like as critical power relative to fat mass or critical power relative to body mass, just because doing that assumes that the slopes and intercepts are gonna be the same between sexes, which in some of these cases they weren't. So I think that was our rationale, but as far as like going one step further and maybe separating it more, I don't know if we looked at any other options, so yeah, it'd be good to look at that too.
And
So for your NBC quadriceps force.
Yeah.
How close is that joint angle to your MRI measured normal?
Yeah, so that is a consideration for sure. It's 90 degrees. And so in the MRI, it was fully extended.
So there's a number of studies that have looked at patellar moment arm, change the joint angle.
Yeah, I actually
You-
have a,
You've taken like one of their regression equations and applied it using whatever your moment arm was in your group to 90 degrees.
yeah,
So then you could have got a more accurate assessment of what quadriceps force was.
totally. Like we, that's a really good point. We did go through some of like that thought process. This is a review that was done on a bunch of different studies that looked at the patellar tendon moment arm. And the results that they got and the regression equations they got have been very variable. And there have been a few studies that said, in inter-individual differences are relatively preserved across the range of motion. And so comparing between individuals makes a bigger difference than comparing within the same individual across the range of motion. So that gives us some confidence in our results, but yeah, definitely more research is needed to get a better way to estimate that moment arm confidently. But we did try it with one equation. I think I might actually even have a slide for it. So we did try it using an equation from this paper, and the results didn't really, statistically didn't really change.
Did you ever try just manually measuring what you, like from the joint center of rotation to the figure out what the moment arm was manually palpating and compare that to what you calculated in the MRI? So you could do that if you're really close to what the MRI gave you, you could manually measure what it is.
We haven't tried that, but that's a good thought. Thank you.
So I might be misremembering here, but was it that the quad mass was not related to your critical intensity in females, but it was in males?
So critical intensity was in everyone. Here, let me go back.
There's maybe a better way to phrase this, was, I feel like I remember there being a measurement where in females quad mass did not impact it, but in males it did.
Yeah, so that was in W prime and our NBC.
Okay, yeah, so. Do you have any speculation then what that means from a training perspective? Typically, think of traditional strength training to increase MPC, totally. That leads to hypertrophy. In theory, that should lead to a greater MPC, but this would suggest that maybe, is it more of a central drive, or do you have any thoughts on what that means from an applied setting?
Yeah, so it is very well documented that like when you hypertrophy a muscle, your MBC goes up. And so we like to think that the samples we recruit are perfectly representative of the population, and in some cases they may not be. So I wouldn't tell someone that, you know, if they trained and their muscles grew in size with hypertrophy and they added more contractile material, that their MVC wouldn't change. I think to get to that point, this study was purely like, observational. And so I think to get to that point, you would need to kind of implement some kind of strength training intervention designed to induce that hypertrophy and look at how it changes, from before to after, to be able to make those kind of recommendations.
Thank you.
Yeah, Michael. Thank you for the chat. This question might be a little naive, but I'm curious. You mentioned that you used the NIRS derived oxidative capacity, which is a non-invasive method to look at that. I'm curious and you got that from hemoglobin mass. What about like looking at blood biomarkers like blood lactate? Are there positives, negatives to either or? And then why did you go ahead with the method that you guys?
So blood lactate, like, or in terms of like during, like at critical power kind of thing. So, yeah, so. Blood lactate, I think, might be getting into a little bit of a different way to estimate that threshold between the heavy and severe domains. So there is another method, the MLSS, the maximal lactate steady state. So that's kind of done with different determination testing. You come in, do a 30-minute trial and look at how much the blood lactate changes from minute 10 to minute 30, and the highest power output where that blood lactate remains stable or relatively stable is said to be your maximal lactate steady state or the boundary. And so I guess it just comes down to a different way to determine it. And so far in the literature, the critical power just seems to represent a better, it seems to be a better representation of that boundary. Does that kind of answer your question? I'm not sure if I got it. Okay.
Why don't we use the term lactate threshold anymore?
Lactate threshold?
Yeah.
That, I'm not 100% sure. I think lactate threshold, I think lactate threshold is kind of ambiguous for me personally, just because lactate threshold often refers to the first ventilatory threshold where lactate increases above resting levels. But in some cases, it's also used as that second threshold where it's referring to lactate becoming unstable. So I'm not sure if that has something to do with it. That's purely might be guessing.
So we still use the term,
Yeah.
not for that.
But maximal lactate steady state just seems like a longer way to say lactate threshold. I'm learning that right now.
Yeah.
And I always thought ventilation thresholds were not lactate thresholds. They were just surrogate markers of when lactate starts to, I always thought, I remember being taught, but I could be remembering this wrong, that your lactate threshold was like, it was related to an exercise intensity that you could kind of theoretically sustain for a long amount of time. That sounds a lot like maximum lactate steady state.
So the key for, I think, understanding why we don't use a threshold for lactate is that the lactate concentration where you can maintain a steady state is kind of an individual value. So you might be able to maintain steady state at a 3 millimole lactate, and I can maintain it 7 or 8. And it doesn't mean I'm fitter, it probably means I eat more carbs. And so the absolute values across people don't mean anything. And so historically, we've tested for this kind of hyperbolic relationship between lactate and intensity. And it's really difficult to turn that value in a test where power is going up into a constant intensity power and just kind of align them. And so people still do a graded test to find a lactate threshold, but it's not synonymous with critical power, and critical power has a physiological basis. So it's probably okay in some situations, but it's kind of a guess at critical power. We know it doesn't get it right every time. It's like roughly in the same area.
Other questions for Rachel? Please.
Yeah, I actually have a question for Martin,
Yeah.
but a comment. I really appreciated your introduction. I mean, and I haven't read your paper, but kudos for that. even though, you know, people have been trying to suppress this for like however many decades. When you walk people through sex differences and you say, here's absolute differences, this is what we get when we normalize by body weight, this is why it's inappropriate, this is why we have to normalize for lean mass. Like, it's obvious, people are like, yeah, I get it, but
That's why I covered that part.
which is really good, but the pushback is not everybody has a DEXA scanner. And so that's why it's been so pervasive in the literature,
Yeah.
just to measure per body mass. So in your paper, did you provide suggestions, like, should we be at least using an in-body instead of a DEXA? Or should we use some like allometric scaling for body mass instead if we don't have?
So I think where we came down was, you need some reliable way to quantify fat mass. And we didn't review all the ways to do it, but you need some way. If you don't have that, you can just measure body mass. And like I said, you can have males 6-7% greater, and you're probably matched. Like as a reviewer, I could accept a certain difference that is probably matched. And trying to match on body mass should be avoided. What's problematic in some studies, so some studies will do that, and when they match them for body mass VO2 max, you're like, okay, so we know the females were fitter, and then you can adjust your interpretation. But the Ansdale data that Rachel showed for the critical torque, they didn't measure VO2 max, so we don't know if the females are fitter. And so in the paper, we come down hard on, if you're not gonna measure it, don't do it. Like it just clouds the literature. So try to measure fat mass. If you can't, let males be fitter, and that's probably okay. But I think to do the research very well, you need to be able to quantify fat mass. Like if that's your research, you just find a way.
Can I ask for your personal opinions on in-body as it's like the biological gyms that like are now pervasive in all gyms, and you can hop on them really quick. Like, I know the limitations, but what would you suggest?
I don't have an informed opinion on them. I need to be convinced that they were okay. I think I'd rather just take VO2 max for body mass and take a 7% difference.
Why not skin calipers? That just introduces more error?
You have to be pretty well trained. Yeah,
Or
it's
But
kind of obsolete,
But
but
here's a new avenue, because we have like a bioimpedance thing in the DEXA room. We could like do both and.
yeah, and I mean, someone probably has.
Yeah.
I'm lucky enough to have a DEXA on the 3rd floor. And so that's been really valuable for all of our research. But I could look up the BIA first DEXA. Certainly like you could do a whole body MRI and other
Yeah, that's
expensive
probably
things.
more.
Yeah.
I have to assume too, there's differences in quality in data.
But yeah, there are, it seems like that would be the easiest alternative to get your meat mass. Do I have a question here?
Just to build on that comment around the MRI a little bit, Martin, I know there's been a lot of pushback on how good DEXA really is at quantifying lean mass proper. Are you aware of any research looking at these relationships and sex differences when we use maybe more exact, precise measurements like something like an MRI or CT?
I think if you're saying lean mass, so DEXA spits out a lean mass, but it's a bit complicated. We really just want to remove fat. I think with DEXA, the next level you could get that could be helpful is muscle mass.
Yeah.
Which we can't get from DEXA, like we can't separate.
Organs.
Yeah, so like that could do something. Ultimately, those are really expensive, complicated methods, and to make these studies possible, I don't think they're necessary, but they would be interesting to see if, like, and that's what we tried to do here was muscle mass of the muscle we're using.
Yes.
We just haven't gone out to whole body or to say these muscles are used in cycling and these ones aren't. Gets complicated.
Andrew?
So I guess to follow up on what Mark just says, did you take the DEXA data and just isolate a region of interest? Would you just do the quad? Like draw a box around your quadriceps on it and be like, what's my lean mass? And see if that mass matched your MRI data.
We didn't do just the quad, but we did the whole like dominant leg. And it was relatively close, like it was relatively correlated. But we did see that, if I remember this correctly, that males had a higher when we looked at like, MRI quad mass relative to DEXA leg fat-free mass, males had a higher proportion than females, significantly. So maybe,
Or proportion on the MRI or something.
yeah, so looking at like MRI quad mass relative to DEXA fat-free mass, that number was higher for males than for females.
So in the MRI data, did you move fat mass from the muscle? Or did you just take the whole volume?
We just took the whole volume,
Yeah, so the difference in DEXA is we can't remove the hamstring.
yeah.
So
Oh,
in
yeah,
the-
yeah, no,
yeah.
but I'm just saying, like, in your quads, if you're saying the mass is higher on MRI. You also have other,
Cause like, yeah, with the MRI, we're looking, obviously there would be some like intramuscular fat in there, but the main sex difference in body fat percentage and that fat content is in the subcutaneous fat, which we wouldn't have captured in the quad volume. So the intramuscular volume in young, healthy adults shouldn't be very different between males and females.
Erin, you had a question?
I understand normalizing to fat-free mass during like unloaded movement. Is it still appropriate to normalize to fat-free mass during like a weight-bearing task like running?
Sorry, can you repeat the question?
Like when you're unloaded or you're not weight-bearing, I understand normalizing and like taking out fat. But when you're running, that adds extra load. Is it still appropriate to normalize and remove the fat mass?
I think just getting back to Martin's point, the idea of it is looking at like the mass that's metabolically active, so the mass that's contributing to your VO2 max. And like that study with the weight vest said, that extra weight is not contributing. It's definitely changing, like for running, it's definitely changing your economy and making you less efficient, but it's not contributing to your VO2 max. So when we're looking at fitness, that's what we care about, is the mass that is contributing. Hopefully that answers the question.
Yeah, I think just to build on that, we're really looking at cardiovascular capacity when we're trying to do this. So if we're talking about performance, like, we're not saying people with the same VO2 max per fat-free mass are equally good at running. For sure, every extra pound has a negative influence on running. So you're right, we're just trying to separate the cardiovascular system's ability to take in, transport, utilize oxygen, and not worry about performance. You wouldn't take runners and rank their fitness by VO2 max per fat. Like it wouldn't help.
Any other questions?
Yeah. Just starting out with some basics, so what is critical aerobic power? When we calculate that as a percentage and you get 45%, what's in the numerator and what's in the denominator?
Sorry, but
Rachel study, maybe?
Yeah.
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 Sorry, so there's two Rachels from that didn't make sense.
No, just in your study, when you talk about critical power, you have something in the numerator and then you divide it, you normalize it by something else, and I'm not 100%
oh,
clear
in this study.
in this study.
Okay, so there was no, there was no normalization. We just used ANCOVA to statistically correct for the variability in the dependent variable based on the covariate. So there was no numerator and denominator.
So your 45% comes from,
45%.
I think there might be like Ansdell and McDougall have it as a percent.
so you didn't have it as a percent.
I don't know,
So in those ones, we're
like
taking,
these ones?
I was just, yeah, you showed slides, there were some
Yeah.
slides where critical power was given as a percentage.
These ones?
yeah, the bottom right.
For example, yeah, exactly.
Okay, so yeah, this was a previous study in our lab, and so
Oh,
this was
sorry, I
looking
thought
at.
it was you, yeah.
Yeah, this was a previous study and this was looking at critical torque, so that's a isometric movement. And so we looked at it as a percentage of the torque at the maximal voluntary contraction. So, yeah, the critical torque, the torque percentage of their maximal
And
torque.
so then you divide by a maximum isometric torque.
Yeah.
But your movement that you do is dynamic.
So this one is isometric. The one in my study was dynamic.
And so in your study, then you have that pedal where you, that machinery. I was very intrigued because you talked about, men being maybe taller and women being shorter and so on.
Yeah.
So if I sit on that machine and I'm tall and the whole is smaller, when we do that and we exert exactly the same moment, would we actually do the same amount of work? Or in other words, would we go through the same knee angle excursion, or would there be a difference between, would a smaller or a taller person actually performing more work, even though the moment is exactly the same?
Yeah, so if the moment was the same, then the person with the longer lower leg would be exerting more external work on the machine. Just because of that longer moment. Or moment.
No, the moment at the knee is the
The
same.
moment at the knee is the same.
So the
So-
force would not necessarily be the same depending on where you measure it.
Right.
But if you in a short or a long person, you could make the moment arm where you measure, you could make that the same amount. So the question really is, do you go through the same angular excursion? Because the work would then be the moment that the work that you produce at the joint will be the moment multiplied by the angular excursion.
Right.
And I wonder if a shorter or a longer person would actually have a different one. So
Yeah, I'm not entirely sure. We are looking into like the ways to, incorporate more like inter-individual differences, like in terms of the effect of gravity and inertia. And we used an electric goniometer to measure knee angle throughout the range of motion. So we're hoping to analyze some of that data going forward.
you
and add to it.
have those measurements, but you haven't looked at them.
We have them, yeah.
Because I wonder if there was differences between a shorter and a taller person in the angular excursion, and you get exactly the same moment profile, then somebody might actually perform more work because
Yeah.
of a bigger angular excursion. And I was just wondering how that might. The other thing is interesting in terms of moment arm that we're looking at, because you mentioned the one thing about the moment arm, and that is that for a given force with a bigger moment arm, you have more torque. But at the same time, if you have two people that are the same height and stature and everything, but their moment arm differs by a centimeter, then the shortening velocity of the muscle would be smaller for the person with the shorter moment arm. And from a force velocity relationship, that would be a tremendous advantage. Maybe one that outdoes the increased moment arm that gives you more torque.
Yeah, that's a good point.
There was an interesting study by Sabrina Lee, who John probably knows, that she was looking at moment arms in sprinters. She had the feeling that the Achilles tendon moment arms in sprinters would be bigger because they would then have more force for a given amount, more moment for a given amount of force. But she actually found that they were significantly smaller in elite sprinters.
Yeah, that's
And
interesting.
the argument there, for me at least, was because she initially said, well, I don't really understand that. And my argument was, well, I think that might be because then you have a smaller shortening velocity, and that force velocity effect might actually offset. So it might be better for sprinting. It might be better for your exercise, a repetitive exercise. When you do it dynamically, their force velocity plays a role. You might actually have an advantage that you have a short moment on rather than big one.
Yeah, that's an interesting thought. I think like as far as, we hadn't really considered that, so yeah, thank you. But as far as trying to, account for any differences in velocity, we made sure cadence was fixed for all of the determination trials for critical power, so everyone was always kicking at 40 RPM. Whether that was the same, angular velocity of the kick, I'm not 100% sure, but yeah, that's a really good thought.
So when you do that with different sized people, you have a different moment arm.
Yeah.
You don't put it 30 centimeters away from from the knee, and it would be the same for everybody, independent of how long the leg is.
Right, no, it's at the foot. So the only thing we change is, like based on femur length, we can move the chair forward and back so that the knee is always in the same position. That's the only thing that changes.
Got it. I think with that, let's thank Rachel for a great talk and